worksheet. First question is, how do carbon dioxide and oxygen move into and out of the mesosphere? Firstly, what is the
Um, your voice isn't clear. I'm pretty sure. I think so.
Sorry, my microphone was.
Okay, now it's good. Okay.
Can you hear me now?
Yeah, now it's good.
Yeah, okay. I was asking that what is a mesophil cell? In the first question, they have asked, how do carbon dioxide and oxygen move into and out of a mesophil cell? What is a mesophil cell?
Like a mesophyll cell is basically like what's it called? It's like a cell that's like in the middle layer of leaves.
Yes, yes, exactly.
Yeah.
So they are asking how do carbon dioxide and oxygen get exchanged?
It's by using what's it called like diffusion, so it spreads around equally.
Yes, the process is diffusion. Okay.
Yeah.
So, and what is diffusion?
Diffusion is like where the particles moves around and like it stops like it's no what's it called diffusion is like basically where like the movement of particles from high concentration to lower and when they're evenly like mixed,
Until they are evenly distributed.
mixed,
Okay.
yeah, or
Yes. Second question is, what are the features of active transport? First, tell me what is active transport?
um active transport is
It is
basically like,
opposite of diffusion. Now you can tell me.
yeah,
Oh,
like it goes from lower concentration to higher concentration.
yes. And you need energy for this.
Yeah.
Which option is correct?
C.
It occurs through the cell membrane. Yes. Particles move from higher concentration to lower concentration. No. Uses energy from respiration.
Respiration.
And what is cellular respiration?
Cellular respiration is like when cells like regrow.
No, respiration. That is cellular respiration means how cells get energy from glucose. You remember we
uh,
have done that glucose
yeah,
is
yeah.
broken
Hmm.
down, then we get
Yeah.
ATPs, right?
Yeah.
Okay, this is correct. C is correct. Okay. Third, a student decided to investigate the effect of different sugar solutions on the mass of chicken eggs. Okay. They used the following methods. Five eggs were placed in hydrochloric acid for 24 hours to dissolve the eggshell. The mass of each egg was measured and recorded. Five beakers were set up. Okay, this is the concentration. And an egg was placed in each beaker for 1 hour. The egg were removed and dried with the paper towel. The percentage change in mass of each was calculated. Percentage change of mass, concentration of sugar. Using information, describe the result in each beaker. Okay, firstly, these are the, on this side, they have mentioned the percentage changes, right?
Yeah.
And the mass. So initially it was more change, and afterwards the percentage change is very less, right?
Hmm.
And the concentration of sugar solution is increasing. So this point, if we take this point, it means at this point the concentration of sugar solution is the
Like
last point.
concentration.
Concentration will be high or less?
Yeah, it will be like increasing, and the percentage
Increase,
of mass will decrease.
decrease. It means when the percentage of mass is decreasing, the concentration of
Mhm.
sugar solution is high, right?
Mhm.
So this is our observation for this, that we can say that with the increase in percentage of the percentage change in the mass of the egg, the concentration is less. Okay, if you see this point, the first point, the percentage change in mass is very high, right? But on the same time, the concentration is zero, right? The concentration is
Yeah.
zero. You see, this is
Zero.
zero. But after some time, what is happening? The percentage is decreasing and the
Hmm.
concentration is increasing. Okay. And in the last, the concentration of the sugar is high. It means the concentration is will be high when the percentage is less. Okay, percentage change is less.
Mm-hmm.
Are you clear with this?
Yeah.
Okay. Use the graph to identify the concentration fluctuation where the mass of the egg would not change. Okay, in which the mass is not changing. Which is the
I was
they
I
have
wasn't
asked.
sure. Yeah, I wasn't sure about B or like the I and II.
Okay, firstly they have mentioned this is just like maths, simple.
Mm-hmm.
We don't need to go into its biology. Okay, they are saying identify that concentration at which the mass of egg will not change. Okay, mass
Mm-hmm.
is constant. Okay, so at what point mass is constant? What do you think?
Uh, I
At
think
what point mass is constant?
at 0 point
Yeah, like
like
this point, right?
yeah, yes, that's
So
one
if
0
we
point.
like this over here, we
Mm-hmm.
get this. So this will be, if we say this concentration will be what? 0 0.2, 0 0.4, it will be approximately 0 0.3, right?
Yeah, like
0.
0.29.
Yeah, 0.29 mole per dm cube. Clear?
Mm-hmm.
Explain why the mass of egg would not change at the concentration identified at part 1. Yeah, at this why the mass percentage will not change.
Maybe because of the like concentration of sugar.
No, otherwise we are saying the concentration is increasing, isn't it, afterwards?
Yeah,
So
yeah.
why at this point the percentage is not changing? Hmm?
Mm.
Like percentage change in mass. What is meant by percentage change in mass? Like what do you understand by this?
Like how many like what percentage will it change?
Yes.
Like how much percentage will it change from it?
Hmm. Yes. Like the why the mass of the egg would not change. So what can we say?
Like I'm not sure.
Any idea?
Is it gonna be because like the water potential is like more inside the egg, and then like the solution is equal, like it has the equal from outside and the inside. That's why it won't change, because if it has both, so
Yes, exactly.
like there's no movement of water.
Yeah, you remember we have done these terminologies hypertonic, hypotonic, and isotonic solutions.
Yes, like you
Yeah,
told me, when
like
some
yes.
water is inside and some
Hmm.
isn't.
Yeah.
And then
Isotonic
one,
means yes, yes.
yeah, isotonic is, I think, like it has water inside and outside.
The same concentration. The concentration of water
Yes.
is same outside and inside. So this is the point where the solution is isotonic. Okay, where the egg
Yeah.
has equal concentration of water inside and outside. That's why at this point the percentage is not changing. Okay. Otherwise,
Yeah.
what
And
are the other two conditions? Hypertonic and hypotonic.
hypertonic has like more water like outside the cell
Hmm.
and
And
less water inside the cell, and
yes, and inside there is
ha
more salt. So what will happen? The water will
uh
go inside, right?
yeah, it'll go inside.
And what about hypotonic?
Hypotonic is like there's less uh
Hmm.
there's
Hypotonic
what's it
cell.
called?
Hypotonic cell means
Yeah,
there
there
is
will be
more
like
water inside,
inside, and then there will be less water outside.
so water will move outside, right?
Yes.
Okay, good. Yes. What are the explain the result that were obtained in the beaker with a concentration of 0.8 mole per dm cube? Concentration of 0.8 mole per dm cube. This is 0.8 mole per dm cube, right?
Yeah.
So what will be the percentage? We can take this line over here, this one,
Yeah.
right? So this will be what?
Yes.
Minus
Like
15, minus 10, minus 11, minus 12, minus 13, right?
13. Yeah,
Yeah,
13.
minus
Minus 13.
13%.
Yeah.
Okay. Diffusion and osmosis are the two ways in which molecules can move. Compare diffusion and osmosis with
Yeah,
each other.
I
Mm-hmm.
just wrote like similarities like and differences on a table. The similarities are like they both involve particle like moving down a concentration gradient like high to low.
Mm.
Yeah, and I wrote like both are passive process that not require ATP solution, won't require energy.
Yes. Like
Yeah, and
in diffusion, we don't particularly talk about the water, right? But in
yeah,
osmosis, we talk about the water. Okay, like the solvent and yes, and what you are saying?
like we don't talk about like energy and like ATP because it doesn't require it.
Hmm. And second thing is for diffusion, we don't need to have a membrane, right? Diffusion can
Yes.
occur of where there is high concentration, and the particles can move from that to the area where there is low concentration. But when we talk about the osmosis, osmosis require a partial membrane. Okay, it requires a membrane. You remember
Hmm.
we did the uh, when we were doing the osmosis, I told you like there is big glucose molecules, right?
Yeah.
And small water molecules. So there is a membrane, this membrane, right?
Hmm.
So over here there is more water, right? But
Hmm.
over
Yeah.
here there is less water. So what will happen? The the water will move from here to this side, okay,
Yes.
from left to right
Yeah.
side. So this is
Yeah.
what this is known as osmosis. For osmosis, we need membrane, okay? We need membrane,
A
partial,
partition. Yeah.
partially permeable membrane, right?
Yeah. Where
Partial,
proteins can't
yes, yes,
go, only some substances can go, like
yes,
water,
exactly. Yes, and the small
sugar.
substances, yeah, sugar
Yeah.
molecules. It depends on the cell membrane, okay? Like the proteins cannot
Yes.
move because proteins are their size is too much large. That's why they cannot pass. Okay, so these are the differences. Good. Next, we have to label these things. What are these things? Outside, there is
The
A.
the
What is
first
A?
one, A is the cell wall.
Yeah, this is cell wall. Okay.
B is like the chloroplast. There's a lot of
Yeah,
chloroplast. And
chloroplast. Yes.
then C is
Hmm.
like it was. I wasn't like what's it called like
It is a vacuole, large vacuole.
yeah,
In the
vacuole.
plant
Yeah.
cell, there is a large vacuole.
Yeah. Yeah.
What's D?
D nucleus.
Yeah, this is nucleus. What is E?
E is cell membrane.
Yeah, cell membrane. Good. Okay. The box on the left contains the letters that identify some part. The box on the right shows some functions of some part. Like these. We have to match, right? Oh,
Mhm. Yeah.
A was what? A was cell wall, right? So what's the
Yeah.
function of cell wall?
The function of what's it called? Cell wall is um like strengthen the cell.
Strengthen the cell. Okay. What about B? B is okay. What was B? Firstly, B was chloroplast. Yeah.
Yeah, like where photosynthesis occurs.
Okay. C is vacuole.
Vacuole. Yeah,
Hmm.
it's like where like contains the DNA genetic material.
No, no, no. That is in nucleus.
Oh yeah, nucleus. Sorry, sorry. Does it like transport the nerve impulses
No.
filled
You
with
know what?
sap to
Yes,
support
yes.
cell?
I told you that the vacuole, if vacuole is filled with water, it makes the leaf turgid. Okay, the leaves are turgid. But
Mm-hmm.
if the vacuole is empty, like there is less water in the vacuole, then we say the leaves are flaccid. You remember?
Mm-hmm. Yeah,
So it support the cell. It is filled with the sap. Sap is like liquid in this. Just like
yeah,
the in the animal cell there is cytoplasm.
yeah.
In
Mm-hmm.
the plant cell
Yeah,
there is cytoplasm as well, but the sap is like a liquid. Okay, it's a liquid material. Okay.
yeah,
Uh, what about D? D is nucleus.
yeah. Nucleus is where it controls the genetic material.
Yes.
And
E
E
was
is
cell
like
membrane.
where
Hmm.
cell membrane portion, yes,
Yeah.
where it controls the substances and
Yes.
leaves.
Okay. Good. Mm. State the name of two different types of plant cells. Mm.
Root hair cell and palisade cell.
Yes. Root hair and palisade. Okay. Next, a student used a potato as a source of plant tissue. The student cut six cylinder-shaped pieces of the potato. Each potato cylinder has the same diameter. Each potato cylinder was immersed in either water or one of the five different concentration. The student measured the length of potato cylinder before immersion and after immersion. Okay, 30 minutes after that. What are the results? These are the results. Use the information and calculate the change in length of the potato cylinder immersed in zero point 0.8 meter. Okay, now we have to calculate this, right? How can we do
Mm-hmm.
this? Did you attempt it this?
No,
Did
I
you?
didn't do C.
Okay. What can we do? What's your idea?
I think like to calculate like the potato cylinder, like you have to calculate like all of the like
Hmm?
moles and stuff.
We need to check. No, there is nothing to do with moles. Basically,
Hmm.
at yeah, in this they can. What is this? They have uh already they have mentioned mole per decimeter cube, right? 0.0 mole, zero
Hmm.
point 0.2 mole, 0.4 mole, zero point 0.6. If we see this, so the potato cylinder, firstly before immersion was this, okay? After immersion, what happened? This is after immersion, this was 49. After immersion, it is
Hmm.
52. It means it has gained length, right? The length is increased.
Yeah, yeah.
And in the same, in this as well, 50, it was initially 50, then it became 52. So in this length is also increased. What is this telling us? That water potential outside the potato cell was higher, okay? That's why now water
Hmm.
is entered in this, okay? And
Hmm.
when the water entered in it, the cell become turgid, right?
Hmm.
Okay, these are the first two. Then we can check the last two first. Okay, then the
Yeah,
last 0.8. If we see the trend in the last, if you see the last, there is 50.
yeah,
Okay, the 50 both have
yeah.
50 before and afterwards it is decreased, right? 48.5. It means
Hmm.
there is a change of minus 1.5, right?
Like I think it's like the like cylinder before immersion minus
Yes, yes,
cylinder after immersion.
immersion. Yeah.
Yeah.
So
So
this
it
is,
will be +1.
yeah, it will be plus one. Just I am just telling you that what is happening in the first
Hmm.
two, the in every cell there is osmosis happening, right? But
Yeah.
if we talk about in the first two, osmosis is happening in a way that water is entering, okay? In these, water is entering in all these. If we talk about water is entering in them, but in these, water is leaving, right? Are
Yeah.
you getting this?
Yeah.
That in all these, because the length is increased, so it means water entered inside.
Hmm.
That's why length increase. In this, water is removed from the cell, okay, from the original cell. Clear?
Hmm. Yeah.
In all of these, osmosis is happening. Use the information, calculate the percentage increase in length of the potato cylinder emerged in zero point 0.2 mole. Okay, this one, zero point 0.2 mole. What is the percentage increase in length? Okay. How can we calculate the percentage increase?
Percentage like increase. You have to
Hmm?
wait. What's it called? Like
Yes.
wait.
You know what
You
is
have to like yeah, minus the immersion before and after.
it?
Yeah.
That will be difference. How can you get a percentage from that?
Oh, oh, I thought we were
How can
doing
we
like we were
hmm?
calculate. Okay, no, no.
Yeah,
Okay,
yeah.
so sorry, sir.
Change
I
in
thought we
length?
were doing I. So,
Firstly, what we will do, we will take the, no problem, no problem. Take it
yeah.
easy. What is the change in length? They
It's,
are talking about the zero point 0.2, right? 0.2.
yeah,
Change in length is 2, right? Plus 2.
yeah,
What is the initial original length?
50.
50. So it means
Yeah.
2 divided by 50 multiplied with 100, right?
Multiply by 100. Yeah.
Yeah, exactly. You get this?
So that's gonna be 4%. Yeah.
Yeah, 4%. Okay. In every section, in every sugar solution, we will do the same. Okay. If
Hmm.
they ask you what is the percentage, you need to check the difference. What is the difference? And what is the initial value? Okay. What is the initial length? Divide
Hmm.
that difference with the initial value, multiply with 100. Clear?
Yeah.
This was very easy. This was, I was expecting that you must have attempted this.
No, it's not like I didn't
Uh,
like I understood now, but like it was
uh,
written differently.
hmm. Okay,
Yeah.
okay. No problem. Sometimes, it looks like it is a very big data and we are afraid of
Yeah.
this. But this is very easy. You see, you just told me this. I didn't told you. You just have to check the trend. What is happening? What is being followed? It's just like maths. Okay. State the name of the process that causes water to enter or leave the potato cells.
Mhm.
Hmm.
So like to do.
Hmm.
Oh, this is what's it called? Wait, I've a process that occurs. Is it like wait, I
Hmm. Very easy, very easy. We have done
yeah,
this
yeah,
before.
just yeah, osmosis. Sorry, by
Yeah,
water. Yeah,
simple. Osmosis,
I've
osmosis. Okay.